introduction to your DNA. Today I'll be mesmerizing your brains on the topic of DNA replication. As you know, DNA is made up of genes. And specifically today, we will be discussing the topic of
unzipping genes.
What is DNA, you might ask? This chain of polynucleotides is what holds our genetic coding. Each acid comes together in a group of 3 to represent an amino acid, which is used to create the many types of proteins in the cell. DNA is formed by a phosphate group, a 5-carbon molecule, and one of the 4 nitrogen bases, which are A for adenine, C for cytosine, G for guanine, and T for thymine. Our genetic material creates a double helix or 2 rows of nucleotide acids which twist into a spiral. The spiral is caused by the hydrogen bonds between the 2 nitrogen bases. Adenine bonds with thymine and cytosine bonds with guanine to create this beautiful structure we know and love. It's the order of these 4 bases that allows your DNA to create you. To create you, the DNA must replicate itself into tons of DNA molecules so that it would be prepared for mitosis. The way the DNA replicates is in a semi-conservative replication process. This process was suggested by Matthew and Franklin in 1958. In this replication, this process begins with the long chains of DNA are wrapped up into a nucleosome by the enzyme histone. This prepares the molecules for becoming a chromosome. In the first step of DNA replication, the double helix unwinds the DNA and unzips as hydrogen bonds are broken by an enzyme called DNA helicase. This unzipping creates a replication fork. A new enzyme, DNA polymerase, is then used across the leading strand, the one that goes in a 5' to 3' direction. The existing polynucleotide acts as a template, while DNA polymerase catalyzes the reaction which causes free nucleotide to move toward the exposed base of DNA. These bases pair up like they are on a dating show and create a new chain of molecules. Enzymes, DNA, ligase, next form covalent bonds between free nucleotides. This connects them all together, creating the chain. On the lagging strand, the nucleotides follow the 3' to 5' direction. Since the polymerase is very specific and wants everything her way, this strand of DNA is replicated differently. RNA primase comes and lays down its primer on the lagging strand. DNA polymerase then comes. However, it does not follow the DNA helicase. It instead moves in the opposite direction, following the 5' to 3' route. This process continues until the entire lagging strand is complete. The replicated DNAs are then removed and turned into long chromosomes. It is essential for growth in every living organism, and knowing it will increase your IB score. Create the many types of proteins in the shell. So, welcome to this episode. Nucleotides. This connects